Thanks, Alyssa, so much for your kind introduction. Also, thank you and the organizers for this awesome webinar series. It's been such a treat to hear the pioneering work in the field and the incredibly exciting vision for the future of targeted protein degradation. I also need to thank my team, starting at St. Jude, where Dave Duda, Danny Scott, with help from Jenny Olszewski and in collaboration with Arno Alpi and Wade Harper's lab, laid the foundation for our more recent studies by PhD students Key Bake and Daniel Horn-Getgo, with help from numerous members of our lab and in collaboration with Gary Klieger, Hybe Ova, my dear late friend, and our cryo-EM facility. I also need to thank numerous sources of fundings and disclose that I'm on the scientific advisory board of Interline Therapeutics. Okay, here I've shown a range of proteins with different structures. And that's because different cullin-RING ligases, ubiquitylate disordered proteins, folded proteins, proteins in complexes, or even proteins that form higher order assemblies like shown in this filament. Today, I'm going to show you the structural mechanisms by which all of these specific proteins in olive are ubiquitylated. And they can be ubiquitylated by cullin-RING ligases because the cullin-RING ligases form a large family of modular multi-protein E3s. So in human, there are 6 cullin-RING complexes that assemble with numerous substrate receptors, more than 250, that ubiquitylate diverse substrates. Today, I'll be talking to you about cullin-RING ligases in the skip1 cull1 F-box protein family that are nucleated by cull1, that in all of my slides will be green. Cull1 partners with the ring protein RBX1 that in all my slides will be blue and on the right. And on the left, in all of my slides, cull1 binds interchangeably to more than 60 different substrate receptors, which are skip1 bound to an F-box protein, which will be shown in a purple color, bound to substrates in an olive color. Now, today I'll be talking to you largely about 3 of these: skip1 BetaTRcp that recruits disordered proteins, I-kappa B alpha and beta-catenin for ubiquitination, skip1 FBXL3 that recruits folded proteins cryptochrome 1 and 2, and skip1 skip2 that binds this protein CKS1 that helps recruit P27, often in complex also with cyclin A CDK2. Now, we've had these structures of different substrate receptors, they're different substrates for many years, but it's been a big question: how do they get ubiquitinated? Now, part of why this is a question is because cullin-RING ligases themselves do not mediate ubiquitination. In order for ubiquitination or any E3 ligase activity to happen, a cullin-RING ligase's ring domain has to partner with another enzyme that mediates ligation. In humans, cullin-RING ligases partner with more than 10 different other enzymes carrying either the ubiquitin-like protein NET8 or ubiquitin. And these different partner enzymes specify different activities. So, for example, the ring domain of RBX1 partners with a NET8E2 that can transfer NET8 specifically to a particular lysine on the cullin, or RBX1 can partner with many ubiquitin-carrying enzymes. Work originally from Pan studying SCFs, and more recently by the Ebert, Chamberlain, and Rolfe groups studying E3 ligases mediating targeted protein degradation, have shown that specific cullin-RING ligases partner with pairs of ubiquitin conjugating enzymes, one that links ubiquitin to a substrate, and then another one that extends a polyubiquitin chain. And we've been interested for many years in how cullin-RING ligases mediate catalysis by partnering with various ubiquitin or NET8 carrying enzymes. Today, I'm going to tell you about 2 distinct mechanisms by which SCFs partner with either an E2 or an E3 to link ubiquitin to a substrate. I'll show you the structures explaining these mechanisms, and then try to highlight why the different mechanisms allow ubiquitinating diverse substrates. Now, you're probably all most familiar with the pathways where cullin-RING ligases utilize an E2, because these are conventional E1, E2, cullin-RING ligase E3 cascades. In the conventional view of ubiquitylation, ubiquitin is activated by an E1 enzyme. Its C-terminus is then transferred to the active site cysteine of an E2 enzyme, generating this covalently linked E2 ubiquitin thioester bonded intermediate that partners with the cullin-RING ligase to transfer ubiquitin to a substrate. Now, several years ago, Danny Scott, along with Dave Duda in my lab, discovered a new mechanism of cullin-RING ligase activity that involves not only an E2 enzyme, but also an RE family RBRE3 enzyme partnering with the cullin-RING ligase, forming an E3-E3 superassembly mediating ubiquitylation of cullin-RING ligase substrates. And the way this works is that ubiquitin is activated by E1 and transferred to an E2, but this E2 doesn't work with cullin-RING ligases. It instead works with this RE family RBRE3. Ubiquitin is transferred from the E2 to the RE family E3, and then via the cullin-RING ligase to the substrate. And I'm going to show you the structures explaining both of these mechanisms. Now, as I said, I think everybody's very familiar with the E2 mechanism because we sort of had a structural view of this that's been in textbooks for almost 2 decades. So work from the Pavlitich lab, this Carey lab, and the Jones lab have shown how various substrate receptors bind to substrates, how they bind to cullins in green, how cullins bind to RBX1 in blue, and how RBX ring domains and how ring domains bind to an E2. And that sort of led to this canonical assembly for cull1 based E3 ligases. And this, of course, eponymous cull2 ligase that was discovered by one of the organizers of the series, Bill Kalin. And more recently, through work of the Thoma and Fisher labs, and also the spectacular structure by Alessio Ciulli, showing for the first time how a PROTAC mediates interactions between a substrate and a cullin-RING ligase, we can even understand how the architecture for targeted protein degradation ligases is achieved. But it doesn't really matter whether it's an endogenous cullin-RING ligase, a cull2 based ligase, or mediating targeted protein degradation. There's been this mystery, a conundrum, because we haven't known how ubiquitin would start here on the active site of the E2 cysteine and get over here to the substrate more than 60 angstroms away. And this question, answering this question, has been a quest, if you will, for me for a couple of decades. And we started by focusing on this E3 ligase here, where the F-box protein is BetaTRcp and it recruits peptide substrates, in part because the ubiquitin carrying enzyme was well validated. It's UBE2D, also known as UBCH5. And this was very nicely validated in cells by Pan, who showed that knocking down UBCH5, also known as UBE2D, stabilizes substrates of SCF beta-TRcp. So Dave Duda, when he joined my lab as a postdoc, set about trying to reconstitute this ubiquitination reaction. He purified SCIP1 bound to beta-TRcp, cullin-1 bound to RBX1, UBE2D. He added a beta-catenin peptide substrate that totally matches the structure. He added E1, ubiquitin, and magnesium and ATP, and mixed them together, and this is what he got. Now, you can imagine how incredibly frustrating this might be for a postdoc to reconstitute this famous ubiquitination reaction, and this is what it would look like. But fortunately, Dave is a very scholarly guy, and he realized that cullins come in a different form, that cullins are often modified by this protein NET8 that's 60% identical to ubiquitin, but it's not ubiquitin. And NET8 C-terminus becomes site-specifically linked to this particular lysine on the cullin. And there were a lot of papers that had suggested that netylation activates cullin-RING ligase activity. And this is just an experiment that was done by Danny Scott in my group. Probably half of you on this webinar have done similar experiments where, thanks to the Millennium Takeda inhibitor, MLN4924, you can inhibit this netylation, strip NET8 off the cullins, and you can see that they migrate only in this singular fast migrating unmodified form, and that concomitantly stabilizes cullin-RING ligase substrates. So Dave set about to ask whether if he adds NET8 to the cullin, does that do anything? So Dave purified enzymatically netylated cullin-RBX1 and used that in his reaction, and voila, this massively stimulated ubiquitylation. Now, I realize massively is a very qualitative term, and fortunately, we have recently collaborated with Gary Klieger, who could put a number on it and show that it really is massive. Netylating the cullin stimulates this ubiquitination reaction by an astounding 2,000-fold. And so we've really wanted to understand how netylated cullin-RING ligases ubiquitinate their substrates. Now, Dave Duda was the first member of my lab to try to work on this, and to understand what NET8 does, he tried to crystallize a netylated cullin-RING ligase. Dave tried to crystallize netylated cull1-RBX1, cull2-RBX1, et cetera. And only when he finally got to the last cullin, cull5-RBX1, and actually only this part of cull5-RBX1, was Dave able to get crystals of a netylated complex. Because there had been no structure of an unnetylated cull5-RBX1, he also had to crystallize the unnetylated counterpart. And since it looked just like this, I'm going to continue to show you the structure of BetaTRcp for the unnetylated cullin. And for the netylated cullin, I'm going to show you a hybrid structure where we've just docked Dave's cull5-RBX1 structure on the right side of the complex, and that's shown here. And so in Dave's crystal, he could see that NET8 linked to the cullin, this part of the complex moved, and the RBX1 ring domain was in a completely different orientation. And because Dave had two different structures within his crystal, he could see that in the netylated complex, the RBX1 ring domain moves. More recently, Ki Baek, a PhD student in my lab, actually determined the structure of this complex, NET8 modified substrate bound SCF BetaTRcp, and indeed his data suggests that NET8 bound to the cullin and the RBX1 ring domain are moving. So with this, we might ask, okay, does this explain how a netylated cullin-RING ligase mediates ubiquitylation? You know, we had these two models. This model here on the left, we know is off, you know, that's kinetically very inactive. And what about this model on the right? Well, if we consider that the substrates of this E3 ligase are disordered, and we take a disordered peptide, and we consider that the ring-bound E2 is now moving around, we can ask, is this really help our model for ubiquitination? Seriously, it seems like the chance of catalytic encounter is still very low when you have a moving substrate and a widely varying, all over the place moving E2. And so we hypothesized that the conformation mediating ubiquitylation may be transiently immobilized under the condition where the substrate lysine was accessing the active site of the E2 ubiquitin intermediate. And that this would actually take place in a rather restricted conformation because the substrate would be bound to BetaTRcp here, and the UBETD ubiquitin intermediate would somehow be bound to the ring and netylated cullin. The only problem is that this is a very fleeting intermediate that rapidly produces substrate. And so in order to try to visualize this, we thought that if we could arrest the reaction by simultaneously linking the substrate and UBE2 ubiquitin complex, that it could capture the mobile netylated cullin-RING ligase in the form that mediates ubiquitylation. And that's the strategy that was taken by Ki Baek, a very talented PhD student in my lab, in collaboration with a brilliant chemist, David Christ, with great help from Rajan Prabhu, who's our manager of structural biology in my new department. And so this is the structure representing ubiquitin transfer from UBE2D to a substrate bound to SCF BetaTRcp. And I could talk about this structure all day. But because it's published, I'm just going to tell you 5 key points that I want you to keep in mind in terms of understanding how cullin-RING ligases ubiquitinate their substrates. The first point is that this structure changes our view of how cullin-RING ligases mediate ubiquitylation. Instead of the E2 ubiquitin active site being way over here and widely separated by a 50 or 60 angstrom gap from where the substrate receptor is, now the E2 is actually adjacent to the substrate receptor And in fact, we could even visualize the peptide in the active site of the E2 here, adjacent to the E2 catalytic cysteine and the C terminus of ubiquitin. The next thing I want to tell you is how this is organized. This involves a catalytic module that is very well characterized, a ring E2 ubiquitin intermediate. And the RBX1 ring in blue bound to UBE2D in cyan and ubiquitin are arranged in the canonical active E2 ubiquitin conformation that was first postulated by Chris Lima and shown in crystal structures and NMR data from Ron Hay and Danny Wong and Rachel Clevitt. And that's just shown here by superimposing the RBX1 ring UBE2D ubiquitin intermediate with a structure from Danny Wong in red. This really is a ring E2 ubiquitin intermediate. But in the context of the whole ubiquitylation complex, it's very precisely placed. And so the third thing I want to tell you is that it is placed by interactions with NET8. NET8 is really the star of the complex. That's why it makes this astounding effect on ubiquitination. And the reason NET8 can do this is it makes 3 interactions. And I think 3 interactions are really important because if you can fix 3 moving parts at 3 sites, then that immobilizes a complex. If only 2 sites are fixed, then there can still be moving around, but the 3 sites really anchors the complex. And the 3 sites were also very important because they actually answer longstanding mysteries in the field of cullin-RING ligase ubiquitylation. So I'll just quickly walk you through them. Mystery 1 we call backside binding, because NET8 binds to the backside of this E2, UBE2D. This was named the backside by Rachel Clevitt because this backside is completely opposite the active site, which is over here. Here's the active site, and here's the backside of the E2. And the reason this was a mystery for cullin-RING ligases is that more than a dozen years ago, it was shown that isolated NET8 binds to isolated UBE2D through this backside interaction, but it had been absolutely impossible to make a model for how NET8 could be linked to the cullin and RBX1's ring domain bound to an E2 and still have these backside bindings. And that's because there was no previous structure that could accommodate this catalytic geometry. But now we see it in the active conformation. The second interaction involves NET8 binding to the domain to which it's covalently linked. And this involves a residue called glutamine 40. And this glutamine 40 in NET8. And the reason glutamine 40 was on our radar was that the Shao, Oswald, and Sasakawa labs had shown that pathogenic bacteria that cause pretty bad infections, as shown here, secrete an effector protein, CIF, that deamidates this glutamine 40. And that would and this ablates cullin-RING ligase ubiquitylation, but nobody knew why. We now recognize that a negative charge at this interface would totally blow up the interaction and it would prevent achieving the catalytic geometry for ubiquitylation. The final mystery we call why NET8. And we say why NET8 because why do cullin-RING ligases have to be modified by a protein that's 60% identical to ubiquitin? I mean, ubiquitin can be transferred in so many different ways. It really was always perplexing to us why you needed a completely different protein to do this. But a lot of residues that differ between NET8 and ubiquitin cluster in this region of NET8, and this makes the third contact with the cullin. And indeed, if key ubiquitylated, made a ubiquitylated SCF, and Gary measured the activity with that, we show that ubiquitin is 60-fold worse than NET8 at activating substrate ubiquitylation. So NET8 coordinates this catalytic geometry, and this catalytic geometry answers many long-standing questions. The fifth thing I want to tell you about this complex is that it's optimized for BetaTRcp because right here, BetaTRcp, the F-box protein, actually contacts UBE2D. And mutations in UBE2D at this interface do have a mild, but nonetheless real effect on ubiquitylation. And so all of these interactions between NET8 and UB2D and the cullin, RBX1, ubiquitin, and BetaTRcp place the active site in a very specific orientation. Now, this orientation is not actually adjacent to the degron. There's still a distance between the part of the peptide that was bound to BetaTRcp and the part that's bound in the active site, and we couldn't see the density for the intervening part of the peptide in the structure. And so this places some limitations on what substrates could be ubiquitinated. If we align all these substrates, known substrates of BetaTRcp by their degrons, shown in yellow, that corresponds to this region, and place black bars at the position that would demark where a substrate lysine would have to be, and then color all the potential lysines red, we can see that all of these substrates have lysines that are in positions that are quite optimally oriented to mediate, to match this catalytic architecture. And in fact, Gary Klieger quantitatively tested this by asking whether substrates with lysines that could be placed distally from the degron would be ubiquitinated, or if he put a lysine that would be approximately here and couldn't reach the active site, what would happen? And he showed that substrates that were too short to match this catalytic geometry, basically ubiquitination was undetectable. So just to summarize, we now have visualized this canonical E1, E2, E3 ubiquitin transfer cascade for SCF beta-TRcp. And we can see how ubiquitin linked to the active site of NE2 is placed to be transferred to cullin-RING ligase SCF beta-TRcp substrates. So this was very exciting, but I also showed you how this structure was really meant to match SCF beta-TRcp. And we wanted to know how many different substrates are ubiquitinated. In fact, for example, how these folded protein substrates are ubiquitinated by SCF FBXL3, or proteins in complex are ubiquitinated by SCIP1, SCIP2. We were particularly dazzled when Ning Zhang solved the structure of FBXL3 bound to cryptochrome 1, this protein here, bound to cryptochrome, this protein here, because this implied a very specific structure. In fact, the ubiquitination sites all cluster on a particular part of cryptochrome, which is here, and we wanted to know how that works. So Danny Scott in the lab tried to reconstitute this, and I'm going to show you his effort to do that. And that led to a partnership with Dave Duda and David Ree and Wade Harper's lab that led us to discover an unprecedented E1, E2, E3, E3 cascade. And I'm going to show you Danny, Dave's, and David's data for this. So first, Danny wanted to reconstitute this ubiquitination of cryptochrome. And as a control, he purified SCF beta-TRcp and had a disordered protein substrate, and SCF with SCIP1 FBXL3 bound to cryptochrome 1. Here's a control reaction where Danny does these assays in pulse-chase format. So he first enzymatically links a fluorescent ubiquitin to the E2 enzyme, then he adds the SCF and a substrate and looks at ubiquitin transfer from the E2 to the substrate. And as you can expect from the structure that I just showed you, with SCF beta-TRcp, ubiquitin is transferred to the substrate, on millisecond timescale. Now here's what happened when he reconstituted it with the F-box protein FBXL3 bound to cryptochrome 1, nothing. And he also got nothing if he let this reaction go, not just for 60 seconds, but for an hour, 2 hours, nothing. Now that we have the structure of SCF beta-TRcp, we can see that ubiquitin bound to the E2 enzyme approaches the substrate bound to TRCP. But if we made a model with cryptochrome, it doesn't matter how we tried to bend or twist the complex, we could never see how the active site of the E2 could approach the lysines that are supposed to be ubiquitinated on cryptochrome. Now, Danny is also a resourceful guy, and he tried everything he could to get this reaction to go. He tried every known component of a cullin-RING ligase reaction. He tried other E2s, CDC34. He tried E2s that aren't even known to work with cullin-RING ligases, and nothing worked. But fortunately, Danny's really good colleagues with Dave Duda, and Dave was studying another protein called Re1, which is an RBR family E3 ligase. And when Danny and Dave added this E2 enzyme, UBCH7 or UB2L3, that Rachel Clevitt showed works specifically with Re1, when ubiquitin is on this and he added Re1 to the reaction, voila, wow, this is really amazing. Cryptochrome is ubiquitinated faster than Danny can pipette, and he can pipette pretty quickly. So how did we know to add Re1 in the first place? Well, Arno Alpe's lab had discovered that Re1 binds to netylated cullin-1 RBX1, and Dave Duda was trying to figure out why. To really get a sense of how broad this regulation is, we collaborated with David Re in Wade Harper's lab, who did proteomics to examine the Re1 interactome. And this is an experiment done with a mutant form of Re1 for technical reasons, but qualitatively similar concepts can be gleaned from the wild type protein. But here with this pull down, David saw that a massive number of F-box proteins, COL2 associated proteins, and COL3 associated proteins associate with Re1. In fact, this is 45% of all of the detectable cullin-1, COL2, and COL3 associated proteins that could be identified from those cells. And this absolutely depended on the interaction with NET8, because if David treated the cells with MLN 492-4 to eliminate netylation, he eliminated all these interactions. So Re1 associates with netylated cullin-RING ligases. This is an experiment by David Re, but Arno Alpe's lab also did experiments like this, where when they knocked down Re1, they could see stabilization of cullin-RING ligase substrates, specifically, particularly phosphorylated P27 and cyclin E, that we have subsequently determined the structural basis for this ubiquitination. So what is this Re1 protein? Re1 is a RBR or ring1 IBR RCAT domain E3. And all RBR family E3s have these three domains, but they also have domains that mediate their specific regulation. And RE family RBR E3s have these two domains, an Ariadne domain and UBA-like domain that mediate autoinhibition of Re1. Re1 is basically a dead enzyme on its own. And it's dead for three reasons. First, the Ariadne domain is blocking the catalase cysteine. Second, Dave crystallized Re1 bound to a stable mimic of an E2 ubiquitin intermediate. And Dave's crystal structure, along with data from Katrin Rittinger and Rachel Clevitt, also related data from Sean Olson, and in the context of a structure from Bernhard Lechtinger, showed that this is really inactive. Re1 bound to an E2 ubiquitin intermediate is inactive because the E2 linked ubiquitin is not properly placed. And in fact, we think it's probably substantially moving around in this intermediate. And Re1 is also autoinhibited because what it's supposed to do is have its active site cysteine, which is shown here in this yellow sphere, pick up ubiquitin from the E2 active site over here, and these are separated by 50 angstroms. So, you know, Danny added Dave's totally inactive Re1 to his reactions, and yet he got them to go. And that's because Re1 is activated by binding to netylated SCFs. And I showed you how this happens when in a cryptochrome reaction. If you added FBXL3 bound to cryptochrome, which is sort of represented here, you could see ubiquitylation. And we wanted to see how broad was the ubiquitylation by Re1. And so we also assembled a series of F-box proteins. This is representing FBXW7 with a long disordered substrate where the lysine is a known ubiquitin acceptor site, what we call a short disordered substrate where a lysine is placed very close to the degron, so very close to the binding region of the F-box protein, and also a ubiquitylated substrate. And Gary Klieger quantified the catalytic efficiency of re-ubiquitinating these. And this is amazingly fast. Ubiquitylation occurs with blinding speed on the millisecond time scale. Gary had to use stop flow, quench flow kinetics to be able to quantify these. Moreover, Re1 ubiquitolates all of these different substrates of different F-box proteins with different architectures with similar catalytic efficiencies varying by only roughly an order of magnitude. Now, what happens if we try this with the traditional mechanism with UBE2D? Well, UBE2D is far less efficient for all of these substrates. But I do want to just mention that UBE2D is still superior with SCF BetaTRcp, which I showed you we think it really works with and it's really optimal for. But how does Re1 handle all of these? Well, work from Danny Scott using about 150, more than 150 purified mutant forms of Re1 and mutant forms of cullin-RING ligases and a hierarchical set of 5 different assays worked out the mechanism. And this mechanism is that ubiquitin starts on E2, and in the presence of a netylated SCF, ubiquitin is transferred to the active site cysteine of Re1. And then ubiquitin is transferred from Re1 to a cullin-RING ligase substrate. Danny showed that this first reaction absolutely depends on NET8, but in his collection of more than 100 mutants, he was able to find one that actually bypasses the need of NET8 for the second reaction. The second reaction requires the SCF, but it can be performed without NET8. And there's a reason why I'm telling you this, and I'll get to that in a minute. So Daniel Horn-Getko, a very talented PhD student in my lab, wanted to know how this looks. And he determined a cryo-EM structures of stable proxies for this intermediate, these intermediates, and for the transition states in the reaction. And in order to capture the transition state, Daniel, David Christ, and Monique developed some activity-based probes to capture this reaction. And Rajan Prabhu, our department expert on structural biology, greatly facilitated the structure determination. So in order to visualize these transition states, we needed to be able to trap these cullin-RING ligases as if they were in action, because as I showed you, this reaction is fast. I showed it happens in 7 seconds, but that was actually, it actually happens on the millisecond time scale. So in order to trap the first reaction, Daniel installed an electrophile between the active site of an E2 enzyme and ubiquitin. This is kind of similar to the activity-based probes that have been developed by Satpal Virdi. We had actually been trying to do this even before Satpal published his probes, but with my move and everything, it took a while to optimize everything and bring it to fruition. But like the Virdi lab probes, this electrophile can then react with downstream components of the pathway. And here I've shown how this reactive E2 probe reacts with Re1 only with wild type Re1 in the presence of netylated cullin, and not with an active site cysteine mutant or other mutants of Re1 that Danny showed were unable to mediate ubiquitylation. And then David Christ and Monique Mulder developed, and Daniel developed several different versions of activity-based probes to try to capture substrate ubiquitylation, but all of these probes install an electrophile between ubiquitin C terminus and the acceptor site on a substrate, and they react with Re1, again, only in the presence of Re1, a cullin-RING ligase. And here it's very important that the substrate receptor of the cullin-RING ligase matches the substrate receptor. So these reactions with the activity-based probe match the requirements for the native reaction. We could also get reaction here using the Re1 mutant that bypasses the need for netylation with just a regular SCF as long as it was assembled with the appropriate substrate receptor. And it was one of those complexes that led to the 3.5 angstrom resolution structure that I'm going to show you. But I just want to say that actually matched the structure of a complex with a netylated, that matched the cryo-EM maps that were obtained at lower resolution with a netylated complex that Daniel also obtained. All right. So in order to visualize this, Daniel assembled complexes with both netylated SCF SCIP2 and netylated SCF FBXW7 and their substrates. Daniel has a lot of data, so I can really only give you a summary of what we think is going on. And for the sake of clarity and brevity, I have to oversimplify quite a lot. But the main points I want to make are, frankly, this is a crazy cool mechanism. I've been working on cullin-RING ligases nearly since they were discovered. And I never would have envisioned that a cullin-RING ligase uses another E3 to mediate ubiquitination. But beyond it being super cool, what I'm also going to show you is that both E3s, Re1 and the netylated SCF, undergo complementary conformational changes that explain how two E3s have specifically evolved to work together to mediate substrate ubiquitination. So I'm just going to highlight those, starting with the SCF. So here is our model for netylated SCF SCIP2 on its own. We're sort of inferring this from the parts. Again, cull1 is in green, RBX1 is in blue. And based on the work from Dave and Key on other cullin-RING ligases, we assume that the RBX1 ring domain and netylated cullin domain that would be here are in motion. And on the left side is the structure that Daniel obtained with cull1 bound to SCIP1, SCIP2, the substrate phosphorylated P27 recruited also to this protein CKS1 and also bound to cyclin A CDK2. Now, in the structure with Re1, the RBX1 ring domain and netylated cull1 domain adopt specific orientations. And in fact, this net8 link cull1 has to extend almost as far as it possibly can away from the cullin. In the unmodified structure, this is a helix, but this helix is not visible. And in fact, it would have to unfold for the cull1 link net8 to be placed here. Here is the structure of auto-inhibited Re1, just to show you how we think things might start out. Bound to the E2, UbE2L3 with ubiquitin links to its active site. Now, to show you what happens when this binds to a netylated cullin, I'm going to morph from this auto-inhibited inactive Re, which is auto-inhibited in 3 ways. Its active site cysteine is blocked, its ubiquitin is not placed appropriately, and the active site of Re1 is 50 angstroms away from that in the E2, and morph to the active conformation. So this is a potential trajectory to the structure that Daniel solved, representing ubiquitin transfer from the E2 UbE2L3 to the active site cysteine on Re1, which is this magenta sphere, when bound to a netylated cullin-RING ligase. This shows how the netylated cullin-RING ligase activates the Re1 RBRE3. And I'm just going to highlight the 3 ways that it does that. First, it has to relieve auto-inhibition of the active site cysteine. Now, this is the auto-inhibited structure, where the active site cysteine is blocked by this domain, this Ariadne auto-inhibited domain. And this involves 3 main residues from the Re1, which I've just highlighted this auto-inhibitory domain in purple and the catalytic domain in pink to distinguish them a little bit. And this involves these 3 residues that are mediating auto-inhibition by binding the active site cysteine of Re1. Now, in the auto-inhibited conformation, this domain, this 4-helix bundle Ariadne domain of Re1 cannot bind cull1 RBX1. It has to undergo a conformational change. I'm going to show you that displaces these 3 auto-inhibitory residues and frees the catalytic cysteine as I show a morph between this auto-inhibited conformation and the active version representing the first reaction that I just showed you. So so this is a morph of showing the conformational change, where when activated, this Ariadne domain from Re1 is bound to cullin-1 RBX1. And so when this otherwise autoinhibitory domain of Re1 is bound, then these autoinhibitory residues face away from where they would otherwise secure the active site cysteine, and this activates the ubiquitination. The second way that ubiquitination is activated is that now Re1 can properly bind to the ubiquitin that's supposed to be transferred. And that actually happens because NET8 binds to this domain over here, which realigns the structure. And I'm just going to show you that NET8 and the cullin domain that it's linked to intercalate into this structure to form a singular domain, if you will, that involves NET8, the cullin, and Re1 that over here presents the ubiquitin bound to the E2 in the active conformation. Now, finally, in order to activate Re1, its active site cysteine, which are these magenta spheres, had to traverse 60 angstroms to pick up ubiquitin linked to the active site cysteine of the E2, which is shown here in cyan. And this happens because when properly presented, ubiquitin's isoleucine 44 patch binds to the sequence here that we call a ubiquitin-guided helix that was previously an autoinhibitory element, now forms a helix that guides the active site to pick up ubiquitin from the E2. Next, of course, we want to know after ubiquitin, we want to know, I need to go back, sorry about this. After ubiquitin is transferred from the E2 to Re1, how does it get transferred to the substrate? Daniel determined the structure of a complex representing, or he obtained cryo-EM data, it's lower resolution, but nonetheless, cryo-EM data of a complex where ubiquitin is linked to the active site of Re1. And this is basically what it looked like. We could see that this part of Re1 is still anchored to the cullin-RING, but NET8 linked to the cullin and the remainder of the Re1 ubiquitin intermediate were not well visible and presumably are moving around. Still, ubiquitin has to be transferred from the E2 to Re1, something like this. After that happens, we think cullin-1 NET8 is mobile, this part of Re1 is mobile, but transiently, ubiquitin C-terminus is linked to Re1. And it's linked there in sort of a very outstretched position through these linkers to the Ariadne domain. And this looks like a very uncomfortable, very strained orientation. It would be held there when ubiquitin is linked to the E2 because the ubiquitin guided helix would bind to ubiquitin that's tethered by the E2. But after ubiquitin is transferred from the E2 to Re1, we think it might look a bit like a slingshot that when released would then traverse the 60 angstroms to form this structure. And this is a cryo-EM structure that Daniel obtained where ubiquitin is linked to Re1, bound to SCIP2, and the cyclin ACDK2 here are modeled. From this cryo-EM data, actually Daniel obtained multiple classes, and if we morph between them, we can see that ubiquitin linked to Re1 is in a very specific orientation, which we think leads to its very high level of reactivity, but there's still motions there that would allow such an active site to accommodate a variety of different substrates recruited to a variety of different F-box proteins. And although I don't have time to show you, that is also explaining the structures that Daniel obtained of this intermediate bound to SCF FBXW7 with its substrates. We don't have a model for after ubiquitin is transferred, but we don't have a structure for after how ubiquitin is transferred, but we can model ubiquitin transfer to a substrate. After that happens, ubiquitin is no longer bound to Re1, and that would give some flexibility to this ubiquitin-guided helix, allowing it to unfold and allowing Re1 to resume the auto-inhibited conformation. So I just want to summarize by saying that we started with this structure of a cullin-RING ligase that we now know is not the active conformation. It plays very important regulatory roles that we're very interested in understanding, but not the conformation mediating ubiquitylation. We know that NET8 is important, and part of what NET8 does is that it sets the netylated cullin-RING ligases are mobile. That allows these mobile domains to be harnessed in different ways by different partner enzymes. In a traditional cascade, an A2 enzyme, which I can show you here, is optimized to present ubiquitin to substrates of BetaTRcp. Here, NET8, linked to the WHB domain, is in one position. But in this other form, netylated cullins can employ a different ubiquitin-carrying enzymes, here NET8 over here, to allow this very reactive active site on Re1 to twist and turn just a little bit to be able to accommodate many different substrates. So what does this mean in terms of its implications? And what does it mean for targeted protein degradation? Well, we started off knowing that the cullin-RING ligase was a system with mix and match substrate receptors, hundreds of these that can recruit different substrates for ubiquitylation. What I've shown you today is that the cullin-RING ligase system also involves a mix and match system of different ubiquitin carrying enzymes, not just E2s, but even Re family RBRE3s that can accommodate these range of different substrates and are recruited to different substrate receptors. So just as different cullin-RING ligases can employ different substrate receptors to recruit different substrates, they employ different ubiquitin carrying enzymes with wildly different mechanisms to ubiquitylate them. Another thing is that orientation matters. How substrates are presented, how the ubiquitin carrying enzymes can access them really matters. And I think that this may help explain why linkers matter so much in PROTACs and why the orientations of substrates of molecular glues have to be presented in a particular way to access a particular different type of ubiquitin carrying enzyme. And finally, the system is robust. Honestly, I think it was far more brilliant than Ray Deshays and Craig Cruz might have ever realized at the time to develop the field around cullin-RING ligases, because as it turns out, there's quite a lot of variation in terms of how substrates can be ubiquitinated. Also, based on Key's mutational analysis, we think a lot, but not all the features of this canonical mechanism are used by IMIDs to mediate ubiquitylation of cereblon substrates. But even if there's even if the system is not ideal, ubiquitylation still happens. And so, I think if you just make a good molecule that can affix your substrate to a receptor with enough time, it seems like some ubiquitin carrying enzyme can modify the substrate. And so with that, I just want to thank again my colleagues. I'm incredibly grateful to have worked with this amazing talent, very adventurous set of scientists and our wonderful collaborators. And I also want to thank biology for such fascinating regulation, because I never in my wildest dreams could envision this. And I'll just stop here and I'll be happy to take questions.